This lecture is the second lecture on the cardiovascular system and covers the heart. So when we talk about the heart, we need to first think about what the heart does to pump blood to different areas of the body. With this image, you see this young man that has lighter blue and lighter red areas that go to the lungs, and then you see darker blue and darker red areas that go to the upper portions of the body and the lower portions of the body. Those are indicating, those different areas, the light colors and the dark colors, are indicating whether they're part of the pulmonary circuit or the systemic circuit. The pulmonary circuit is the lighter colors, and this is when blood goes to and comes out of the lungs. And the systemic circuit is carrying oxygenated blood from the lungs to all areas of the body, except to the lungs. And it's returning the deoxygenated blood and waste products back to the heart. All right. So even though you don't know the different areas of the blood, of the heart at this point, let's go through the steps of the systemic circuit. And before we even do this, let's take this down to the most molecular level that we have looked at. We are looking at oxygen going into cells so that that oxygen can be used in cellular respiration so that we can make ATP through the use of the organelle, the mitochondria. So I want you to think about this as if you're one red blood cell covered with oxygen and traveling to different parts of the body. All right, so from the heart, from actually the left ventricle, oxygen-rich blood is going to be ejected out of the heart. And we use the word ejected for a reason, because it is at a very high pressure. It's the highest pressure you should have in your body. And that blood is ejected out of the left ventricle into the largest artery we have, which is the aorta. And the aorta is what you're seeing right here. All right, so this is your aorta. Coming off the aorta, you have smaller arteries that branch, and it goes to different parts of the body. So this is a very simplified drawing, but you know that you have arteries that are going to go up to your upper torso, to your brain. They're going to go to your arms. It's going to come down here and go to your digestive system, to your skin, to your urinary system, to all the muscles in your legs and your feet, etc. All right? And so this is happening on all sides of your body. All right? All of this blood that is going at high rate. These arteries are going to branch and become smaller and smaller and smaller till they become arterioles. Those arterioles become progressively smaller, and eventually they will form capillaries. And by the time you get to a capillary, the blood pressure has been greatly reduced, whether it's a capillary in your brain or a capillary in your big toe. Even though the brain is closer to your heart, it has still under greatly reduced pressure. All right. Once the blood gets into the capillaries, oxygen goes out of the capillary and into the cells. Carbon dioxide comes in along with other waste, etc. What else comes out of the vessel? We might have nutrients, ions that are coming out. We're picking up excess ions and waste products and carbon dioxide, right? That's all happening in the capillaries. But let's just call it deoxygenated blood at this point. That deoxygenated blood is then going to go up through the venous system, and that's what you're seeing here, the dark blue. And the blood is then going to enter the right atrium, both from the bottom and from the top, which would be the upper torso. So it's both going into the right atrium. That blood then goes from the right atrium to the right ventricle down here. And then that blood is going to be pushed through the pulmonary artery, and it's going to go into the lungs. And this is where the pulmonary artery is going to divide, and it forms what's called the pulmonary capillary region. That's what you're looking at here. We have these tiny vessels that pass the alveoli that are within the lungs, and gas exchange occurs. When you breathe in, you are breathing in oxygen, and you fill these tiny, 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 very fragile balloons in your lungs with oxygen. Those pulmonary capillaries that are in your lungs are on the outside of the alveoli, and oxygen is diffused into the blood, and carbon dioxide is diffused out of the blood back into the alveoli, so that when you blow out or you exhale, you are getting rid of that carbon dioxide. So number 4, oxygen crosses the membrane into the blood while carbon dioxide leaves the blood through the same membrane. We then have the newly oxygenated blood that is going to flow into the pulmonary veins. And that blood comes through the pulmonary veins, and it's going to enter the left atrium and then enter the left ventricle, and then we go through this all over again. So regardless of how we look at this, arteries always take blood away from the heart, and veins always take blood to the heart. Capillaries is the network between the arteries and the veins. Sometimes they're just called exchange vessels. Now let's look at the anatomy of the heart itself. So it is behind your sternum. You have what are called the great vessels, which are the veins and the arteries are at the base. So the top part of the heart is called the base. The pointed tip is called the apex. It is an inverted triangle or inverted pyramid. There is a pericardial sac that surrounds the heart, and it is found between the two pleural cavities, the right and left. Each one of those pleural cavities holds a lung, either the right or the left. There is a central tissue mass that is called the mediastinum, and that divides the thoracic cavity into the two pleural cavities, right? That is where you find the heart, in the mediastinum. As we talked about when we talked about membranes, and this was in AMD2, remember how we have serous membranes, and each serous membrane has 2 layers. You have the parietal portion and you have the visceral portion. The parietal portion lines the cavity, and the visceral portion is the part that actually touches the heart. It's known as the epicardium, right? There is a cavity between the two, and it has pericardial fluid, which is a lubricant, because if you didn't have it, then every time you had a heartbeat, you would have tissue rubbing on tissue, which causes friction. The sac is mostly made up of collagen. If a person has pericarditis, then you'll hear a scratching sound because the surfaces are actually rubbing, because there is an increase in fluid that collects around the heart. So when you think about the pericardial sac, please look at this diagram and think about a fist being pushed into a partially filled balloon. Make it a water balloon if you'd like, but it's partially filled. Your fist is your heart. The part of the balloon that is touching your heart is the visceral pericardium. There is a space that is the pericardial cavity that has a bit of fluid in it. And the part that is the outer wall of the balloon corresponds to the parietal pericardium. When we look at the chambers of the heart, you know that there are 2 ventricles and 2 atria. Singular for atria is atrium. The ventricles are the lower chambers and the atria are the upper chambers. We've already talked about the fact that the left atrium is where you have blood that is returning from the pulmonary circuit. The blood is then going to leave the left atrium and it is going to enter the left ventricle. With a great contracting of the cardiac muscles, that blood from the left ventricle is going to be ejected into the aorta. Let's pause right here and let's look at the colors of the vessels as they enter or interact with the heart. First, let's look at the aorta, right? You see the aortic arch. It is bright red. I also want you to look at the left pulmonary veins, right? Left pulmonary veins. They too are bright red. I would like for you to look at the pulmonary arteries. Those pulmonary arteries are a strange purple color. But then when you look at the superior and inferior vena cava, you can see that they are blue. Why do we have these different colors? Well, remember that I said when we're talking about arteries, the blood is always going away from the heart. And when we look at veins, the blood is always coming to the heart. So some people get confused because they also think that arteries are always red and veins are always blue. And you can't do that in this instance when you're looking at the heart. Colors indicate whether it's oxygenated or deoxygenated. So if it's bright red, it's oxygenated. If it's blue, it's deoxygenated. That purple is indicating that yes, it's deoxygenated, but it's also in an artery, okay? So don't just think that colors are going to help you in regard to the heart. Notice the left pulmonary veins. They're bright red. Why are they bright red? Because the blood is coming from the lungs, and that is where it just became oxygenated. So not only do you have to know whether it's an artery or vein or arterial blood versus venous blood, but in regard to the pulmonary and systemic circuit, you need to understand the colors. If it's bright red, it's oxygenated. If it's blue-ish, purple, that means it's been deoxygenated, all right? Notice on the right-hand side that the right atrium is collecting blood from the superior vena cava and the inferior vena cava. These are the largest veins in your body. The superior vena cava collects blood from your upper body. The inferior vena cava collects blood from your lower body, things below your heart. That blood is going to enter the right atrium. It's completely deoxygenated. It's under incredibly low pressure. Once it enters the right atrium, it is going to then enter the right ventricle. Upon contraction of the ventricle, the blood is then going to be pushed into the pulmonary trunk, and the pulmonary trunk is going to divide into the right and the left pulmonary arteries. That blood is then going to go to the lungs and be oxygenated. It is then going to come back to the heart through the pulmonary veins. These pulmonary veins are then going to allow for the blood to enter the left atrium. The blood will then enter the left ventricle. Upon contraction, The blood in the left ventricle is going to enter the aorta. It is then going to be spread throughout the body to feed and nourish the tissues, pick up waste in the capillaries, and then it's going to become venous blood. So arterial blood is leaving the heart, venous blood is entering the heart in regard to the systemic system. More on the anatomy of the heart. So if we look at the outside of the heart, the coronary sulcus is going to divide the atria and the ventricles. So here is the coronary sulcus, and it has fat in it. The posterior and anterior interventricular sulci we can find right here. And these separate the right and left ventricles and have the blood vessels of the cardiac muscle. The ventricles, if you look at this cross section of the heart, the right ventricle wall is thinner, the left is thicker, the right is pouch-shaped, the atria both are thin-walled, and they have this expandable outer portion that is called the auricle. And auricle, if you remember A-U-R-I or A-U-R, indicates that we are talking about an ear. Well, you know we're not talking about an ear, but it is an ear-shaped flap, and sometimes it's actually called the dog's ear. There are 3 layers to the heart wall. The first layer, or the outermost layer, is the epicardium, also known as the visceral pericardium. It covers the heart. It is a serous membrane, as we've already talked about. In order for it to be a membrane, remember, it has to have at least two different types of tissue. So there is a mesothelium and a relar connective tissue. The second part is the myocardium. Remember, MYO means muscle. And this is the muscular wall of the heart. This myocardium forms the atria and the ventricles. It has concentric layers of cardiac muscle tissue, blood vessels, and nerves. The innermost portion is called the endocardium, and this endocardium lines the chambers, and it extends over different structures such as the valves, the chordae tendineae, and the papillary muscles. Cardiac muscle cells, as we talked about when we talked about muscle cells, are small in size. They are oddly shaped. Think about them like tree branches. They have these branches that have interconnections between the cells that are known as intercalated discs. The intercalated discs allow for each cardiac muscle cell to be interconnected. They are secured by desmosomes. If you remember, back in A&P1, I talked about desmosomes and hemidesmosomes. These are connections between cells that you can think about as if they are like Velcro. It is different fibers that essentially are like the hook and loop of Velcro, so that the cells can be linked together. So they're also linked by gap junctions. Also remember that when we have cells that are beside each other, they can have no space, which would be a tight junction, or they can have wider spaces, which are gap junctions. And these allow for the force of contraction to be conveyed and also allow for the propagation of action potentials. So I've already talked a little bit about the vena cavas. There's the inferior and the superior, and remember this is part of the venous system. The superior vena cava is going to receive blood from the upper body, the head, neck, upper limbs, and chest. The inferior vena cava is going to receive blood from the trunk and the viscera and your lower limbs. Between the atria is something called the interatrial septum that separates the two of them. And then between the ventricles, you have the interventricular septum. And you can see this interventricular septum right here. It's kind of hard to see the interatrial septum. In the right atrium, there is this structure that I would like for you to notice. I'm circling it in green. This structure before birth is called the foramen ovale, which is the oval space or the oval hole. It is an opening that goes between the goes through the interatrial septum. So remember, the interatrial septum goes between the right, it's the divider between the right and the left atria. So the foramen ovale connects the two atria before birth. All right, I want you to just pause here and think. Before birth, where are you getting your oxygen? Are you getting it from your lungs? Are you breathing? No, you're not. You are not breathing in through your nose and bringing oxygen into your body and inflating your lungs and then allowing your blood to pick up all that oxygen. That's not how that works. As a matter of fact, your lungs are flat. They're not even inflated at all. So you have to bypass the pulmonary system before you're born. And this bypass of the pulmonary system occurs because of the foramen ovale. So blood enters the right atrium and it then goes to the left atrium, and then it goes to the left ventricle, and then it is ejected through the body. And you're picking up oxygen because you have fetal hemoglobin, and so you can pick it up from the placenta, right? So you don't need the pulmonary circuit. When you're born, after you take your first few breaths, that foramen ovale should close and it becomes the fossa ovalis. All right, remember, a fossa is an indentation. A foramen is a hole. So that fossa ovalis is where you have that connection between the two atria. Now, sometimes when a child is born, this foramen ovale does not seal or does not seal enough. Years ago, this was life-threatening. These days, it is not a big deal to close that foramen ovale through surgery. I say not a big deal. Surgery on a newborn is a big deal, no matter how you look at it. However, it is a much less frightening thing. If you've ever heard that a child was born with a hole in his or her heart, this could be one of the reasons why the child had a hole in their heart. Another thing I would like for you to notice is something called the coronary sinus, right? So the coronary sinus opens in, so here it is right here. I want you to see it. See it right there. The coronary sinus opens into the right atrium, and this is where the cardiac veins are returning blood. So remember, the cardiac veins and arteries are the veins and the arteries that feed and remove waste and CO2 from the muscle of the heart itself, right? So the blood on the inside is doing nothing for the heart. It still has to go through an arterial and venous system, which are the tiny vessels that feed the cardiac muscle itself. So once the cardiac muscle is fed, we have CO2 and waste that need to go back into the pulmonary system. And so that blood has its own special opening, which is the coronary sinus. All right. Something else I would like for you to see is called the pectinate muscles, right? Pectinate muscles, you can see them right here. They are on the outside atrial wall and also on the inner surface of the right auricle. In the right ventricle, there are structures that are called trabeculae carneae. And these are the muscular ridges on the inside of the right ventricle. Also on the inside of the right ventricle is this guy right here, which is called the moderator band, right? It doesn't look much like much right here, but essentially it's a rubber band that goes from the interventricular septum to the right ventricular wall, right? Moderator band. It allows for coordination of the contractions of the cardiac muscle cells. It's part of the conducting system, and we'll talk a little bit more about it later. Another thing I would like for you to notice are the cusps. All right, so here's one, here's two, and this has been cut away. That would be three. These are the cusps of the tricuspid valve, or the right atrial ventricular valve. Over here, you can only see one cusp of the bicuspid valve. The other one has been cut away. And this is the left AV valve. Notice attached to each cusp are these strings. And these strings are called chordae tendineae. Each of these strings is attached to a special muscle, which is called a papillary muscle. There's another one right here and another one right here. And these papillary muscles, with the attachment of the chordae tendineae, keep the flaps from going backwards into the atria whenever there is a contraction. So I want you to think about this. When there's a ventricular contraction, this whole area is squeezed and it's filled with blood. We have to stop the blood from going back up into the atria. And the way that that happens, or in order to keep these flaps from flipping back up here, you have the chordae tendineae that is attached. Let's talk a little more about the AV valves. On each side, between the atria and ventricles, you have the atrioventricular valves, or the AV valves. They're one-way valves, as I just talked about, and you don't want backflow during contraction. If you have blood going up into your atria, instead of going either into the pulmonary circuit or the systemic circuit, then you're losing out on oxygenated blood if it's systemic, or actually oxygenating blood if it's in the pulmonary circuit. So blood pressure actually closes those cusps during the ventricular contraction. And as I said, the papillary muscle tense and they prevent the valves from swinging back. The right AV, also known as the tricuspid valve, it's the opening from the right atrium to the right ventricle. The left AV is the opening from the left atrium to the left ventricle. That's one type of valve, the AV valves, of which there are two. Now we're going to talk about the second type of valve, and these are called semilunar valves. When the valves are closed, they look like moons. When you look at the individual pieces, they look like portions of moons, right? That's why they're called semilunar. There are two. One of them is between the left ventricle and the aorta, and it is called the aortic semilunar valve. And the pulmonary semilunar valve is between the right ventricle and the pulmonary trunk, and it's called the pulmonary semilunar valve. Both of them have 3 cusps. They're different, though, in that these cusps are cup-shaped when you look at them from a superior view. There is no muscular support. There are no papillary muscles that help or chordae tendineae. When blood is pushed from, and let's just use the systemic circuit for an example, when blood is pushed from the left ventricle into the aorta, that pressure opens those valves. But when the contraction stops, blood actually will fall back, and because it falls back, it fills those cups and it keeps them closed, right? There is a structure I would like for you to notice, and it is called the aortic sinus. Aortic sinus, here it is right here. Notice it's behind this cusp. So it's at the base of the ascending aorta, and it has 2 jobs. One of them is to prevent the cusp from sticking to the wall of the aorta, but it's also the origin of the right and left coronary arteries. So if you wondered how oxygenated blood actually gets to the muscle of the heart, this is the point where it enters the cardiac systemic system, what is necessary to feed the muscle of the heart. If a person has carditis, it is an inflammation of the heart. If a person has carditis, they can get something called VHD, which is valvular heart disease. A way a person can get that is by having rheumatic fever. People still get rheumatic fever, but it is not as common because if a person has a strep infection, then they often go on antibiotics and it keeps them from getting the rheumatic fever. It can still happen though. When a person has rheumatic fever, they will have a high fever and joint pain and a very specific rash. Over half the people that get rheumatic fever will develop carditis, and that causes scar tissue in the muscle of the heart, in the myocardium, and in the heart valves. So a person can get mitral valve prolapse, and when that happens, it keeps the valves, it allows for regurgitation, allows for blood to go back up into the left ventricle upon left atrium upon contraction of the left ventricle. As you see in this image, you can see blood flowing back up into the left atrium as well as into the aorta. But a person may not notice this for 10 or 20 years. It's because the valves calcify and they thicken and they don't work appropriately. Because a person who has this has a heart that is more susceptible to bacterial infections, which can cause endocarditis, which can be life-threatening, a person who has this disease or disorder, or a person who just has a heart murmur, period, may go on something called prophylactic antibiotics prior to having any dental work done. This is because a person is actually more susceptible to a bacterial infection. And if you remember, your mouth is pretty dirty, and anytime you have work done, most of the time you bleed, and so you can have bacteria that enter your circulatory system, which could have detrimental effects on your heart. All right, so I have talked a little bit about coronary circulation, but let's go into it into a little more detail. These are the cardiac arteries and the cardiac veins. And so they're supplying blood to the heart muscle and taking blood away from the heart muscle that also contains CO2 and waste. So we have the right and left coronary arteries. Here's the right coronary artery right here, and here is the left coronary artery right here. So both of these originate at the aortic sinus. Remember I showed you, you have semilunar valves in here, and those semilunar valves have the aortic sinus in relation to their location. So because we have contraction of the left ventricle, this is going to force blood out, and it also forces blood into the right and left coronary artery that have multiple branches that will feed different areas of the heart. The right coronary artery is going to supply blood to the right atrium and the ventricles, and the SA node and the AV nodes, and we'll talk more about that when we talk about the electricity of the heart. It also supplies blood to the backside of the posterior interventricular artery. The left coronary artery is going to supply blood to the left ventricle, the left atrium, and the interventricular septum. So there's two branches of that, the circumflex and the anterior interventricular artery. So you may have heard of a person that had a heart attack in what was called the widowmaker. And so this widowmaker is the left anterior descending branch of the left coronary artery, all right? That is almost 100% fatal because it supplies so much blood to the left ventricular and the interventricular septum. So the interventricular septum, as you see here in just a minute, we'll see here in just a minute, is incredibly important to you actually having the sending of electrical signals to your heart. So the term heart failure means that a person does not have adequate blood flow to the tissues of the heart due to ventricular damage, right? Due to ventricular damage. If you have ventricular damage, then you don't have those massive, strong contractions on the left side, and the smaller, but just as important, ventricular contractions on the right side. Okay? Cardiac veins. We have the great cardiac vein. It's going to drain blood from the artery and go into the coronary sinus. Remember, I showed that that is in the right atrium. And we have the anterior cardiac vein, and that empties right into the right atrium. The posterior cardiac, middle cardiac, and small cardiac veins both empty into the great cardiac vein or into the cardiac sinus. Right, so let's talk about the cardiac cycle. This is what is occurring between start of one heartbeat and the beginning of the next. And a heartbeat includes both contraction and relaxation. So a heartbeat is a single contraction of the heart. When we think about a heartbeat, you may think that the heart contracts, and that's not exactly how it happens. Contractions first start in the atria and then goes to the ventricles. Your cardiac muscle cells are broken down into what is called the conducting system and the contractile system. So the conducting system is like a conductor. It controls what's going on in the heart and it coordinates what's going on in the heart. The contractile system is the worker. It actually produces the contractions. So if you look at this image at the very top, you can see that we have the SA node, very top of the right atrium. Then we have the AV node. It's not labeled right here. So you have this electric stimulus that travels through this node. It's part of the conducting system. It is going to send a charge to the AV node, which is going to generate all of this energy that is going to travel down the interventricular septum and then up the sides of the ventricles, right? And that is going to allow for a contraction to occur. So remember, this is the conducting system, and then we have the contraction. And the first things that contract are your atria because of what's occurring up here. And then as the impulse travels down here, we have contraction of the ventricles, right? So the conducting system has to be engaged first, and then the contracting system is going to engage. All right, so with this conducting system, specialized cardiac muscle cells, we have the SA node, the AV node, and the contracting cells. So here the AV node is actually labeled. This is an automatic initiation, and it's completely involuntary. Notice you're seeing the generation of an action potential with depolarizations, right? So we have a depolarization event at the SA or the sinoatrial node. So before we go into the words on this, I just want you to look at the colors. In the first frame, step 1, no purple. Step 2, see the purple as it begins to spread over the top of the atria. Notice in step 3, the atria are all purple, and notice that there is contraction of those atria. It is, you look at step 2 and look at how this is not concave, and now look here and see how it is concave. So we have, look how this is rounded on the left side, and look here how it is not as concave in step 3. So what you're seeing here is that the electrical impulse, okay, the conduction has been sent throughout the atria. Notice in step 4 where the purple is. This is showing you where the impulse is. The impulse is now in the interventricular septum. So the impulse is no longer up here. It is in the interventricular septum. But what do you see? The atria are still contracted. Once that impulse travels through that interventricular septum and it makes its way up the sides of the ventricles, what do you see here? You see contraction of the ventricles. So my point with going through the colors first is that you can see that the atria contract before the ventricles, that the impulse is generated in the atria, and that the atria are going to contract because of that impulse. The impulse is then going to travel down the interventricular septum so that then we can have contraction of the ventricles based on that impulse. All right, so now let's look at the words. The SA node receives an impulse, and once that happens, the stimulus is going to spread across all of those atrial surfaces, and once it reaches the AV node, which is AV, atrioventricular, it is in the atrium, but it is the AV node. Once it reaches that, look at step 3, then we have atrial contraction beginning. Not only do we have atrial contraction beginning, but that impulse is then going to travel down the interventricular septum through what's called the AV bundle, and I'll show you more of that in just a minute, through that AV bundle and the bundle branches, which are in the interventricular septum. It keeps traveling, and then also notice in step 4 that it hits the moderator band, right? Remember that moderator band is going to help with the contraction on the right side. And it helps with the papillary muscles and the contraction of the papillary muscles. The impulse in step 5 is then distributed by the Purkinje fibers. So the Purkinje fibers are what you see that are no longer in the interventricular septum, but they're also actually in the ventricle walls. These Purkinje fibers are going to distribute that impulse, and when that happens, the ventricular contraction begins. Okay. We're no longer having active atrial contraction, but are the atria still contracted? Yes. Remember that contraction is active and relaxation is passive. Even though we're not having active contraction, the atria are still contracted. Atria are contracted during a portion of the time while the ventricles are also contracted. Okay, that's something super important to remember. Also notice this. Let's go back to step 1. Now let's look at the AV valves, right? Step 1, AV valves are open. Not wide open, but open. Step 2, contraction hasn't begun. We just have the impulse spreading, and the AV valves are still partially open. Now look at step 3. There is full atrial contraction, and what do you see? The AV valves are fully open. What's the purpose of the atrial contraction? It's to force blood into the ventricles. It only makes sense that the AV valves are fully open. Now let's look at step 4. The atria are fully contracted, but the impulse has not reached the Purkinje fibers so that the ventricles can contract. AV valves are fully open. Now when we get to step 5 and we have ventricular contraction, we don't want those AV valves to be open anymore. So this is where we have the cusps and the chordae tendineae and the papillary muscles all become engaged to keep those flaps from going backwards into the atria. And the pressure itself, again, it's like pushing the fluid up. It's actually keeping those cusps closed, right? And I think you can see that pretty well in this picture. Right, SA node, posterior wall of the right atrium. I think you can see it has something called pacemaker cells in there. There's something called the internodal pathway that connects to the AV node, and this begins atrial activation. The AV node is in the floor of the right atrium. Yes, it's called the AV node, but it's in the floor, and it receives the impulse from the SA node. It delays it a bit, and then atrial contraction is going to begin. bundle is in the septum, and it's going to carry the impulse to the right and left bundle branches, which are then going to conduct the impulse to the Purkinje fibers and to the moderator band. The Purkinje fibers are going to allow for the impulse to be distributed throughout the ventricles. Atrial contraction is complete. Remember, the atria are still contracted. This is when the ventricular contraction begins, and the Purkinje fibers are going to distribute that impulse, that stimulus, to the contractile cells, and those contractile cells are going to allow for the contraction of the ventricle. All right. So on this test, you have to know for the process for this piece of material, you have to know pulmonary and systemic blood flow, and you also have to understand when we have contraction, what valves are open, what valves are closed, what part of the heart is contracted. So you have to understand the cardiac cycle. So contraction is called systole or systole, you can say it either way, and relaxation is called diastole or diastole. If you look at a blood pressure, the systolic pressure is the first one, 110, and then the diastolic pressure is diastolic is around 80, and I'll explain how those pressures are derived shortly. So during systole or systole during contraction, of course blood pressure is going to rise in any chamber, right? You're pushing on it. Think about squeezing a balloon. Doesn't that create a great amount of pressure? And it's also going to fall during relaxation. Blood is going to flow from areas of high pressure to low pressure, and this movement is controlled by the contractions and the one-way valves. Okay. So when you look at this image, let's start where it says start, and the green arrows are indicating to you contraction. The black arrows are indicating to you the flow of blood. All right. During atrial systole, atrial contraction, there is blood that is released into the ventricles, all right? When atrial systole ends and atrial diastole begins, okay, so that once contraction ends, then relaxation of the atria begins. But remember, it's not immediate because it is passive. But the impulse has now made it to the interventricular septum and up through the Purkinje fibers, and we're starting ventricular systole, right? So these are the ventricular contractions. They're going to push the AV valves closed, but it's not enough pressure to open the semilunar valves at this point. We've started the contraction, but it's not quite enough. So those semilunar valves in the aorta and in the pulmonary trunk are not open. As we continue through ventricular systole, this is the second phase, you've increased the pressure, and that is going to allow for the semilunar valves to open and the blood is ejected out of the heart. All right, so now we have ventricular diastole. This is the beginning of the relaxation of the ventricles. All right, so look at the atria while this is occurring. You have the atria that are relaxed. You have the ventricles that are beginning to relax. What you have at this point is, look at the atria. You have blood entering the atria. And because the ventricles relaxed, look at the pulmonary trunk and the aorta. The semilunar valves are still closed, but there's still blood that is flowing back down. And remember, those are actually like little cups. Those cusps are like little cups. And so they fill with blood and they keep the semilunar valves closed. All right, now look at step F. This is the last or late stage of ventricular relaxation. All chambers are relaxed. That means blood is passively coming in to the ventricles, and you have blood entering the atria from the pulmonary arteries and, from the pulmonary veins, and you have blood entering from the inferior and superior vena cava. All right, now I want you to look at the colors. There are 4 colors in these circles, all right? Ventricular diastole is peach, ventricular systole is purple. Atrial diastole is green, atrial systole is beige. Look at the relative amount of time the atria are contracting versus the atria relaxing. And again, look at the relative amount of time that the ventricles are contracting versus the time they're relaxing. So if I ask you the question, are atria relaxing during the time ventricles are contracting? The answer is yes. This indicates relaxation. This indicates contraction. Are the atria relaxing during the time the ventricles are relaxing? The answer is yes. Are the atria contracting during the same time that the ventricles are contracting? No. I'm talking about active contraction. Okay, active contraction. Are they contracted? Yes. Is it active? No. Okay, I just went all through that on a small picture, but I'm sorry, but it's the same thing. I just want you to see all of that. But I also want you to really pay attention to these colors, because you're going to see them again right here. All right, atrial relaxation. Remember, the heart spends most of its time in regard to atria, in atrial relaxation. It's a very short amount of time that it is contracted. If you compare the ventricles, they spend much more time relaxed than they do contracted. All right, so these colors correspond with the colors right here. This is what I want you to see. Let's think about what's going on in the left atrium. In the left ventricle. Here we are with this red line, and this is pressure in millimeters of mercury. We're not quite at 0, but we're close. So this is where atrial contraction begins. And if you look at the left atrium, you have blood is ejected into the ventricles, and here we are, atrial systole ends, and the AV valves are closed. And then we watch what occurs with the ventricles. You have ventricle ejection occurs. So you have the ventricle contraction. So you follow it up. This is at its highest pressure, which is 120. This is when it is ejected into the aorta. As we've peaked with the contraction and we begin relaxation, the aortic valves are going to close. And as we continue with the relaxation, we get the AV valves open, and we get all the way down here where there's very little pressure in the heart, which means that we have AV valves open, and we have the filling of the atria from either the pulmonary blood or the systemic blood. Okay, but you can see where we're getting our pressures. The highest is here, and then when you get the AV valve to open, it's around 90. All right, I just talked through all of that. And that. And that one. All right, when we finally get to step 8, that atrial pressure is actually higher than the ventricular pressure because you've got this passive atrial filling, the ventricles are also filling, and you're at the end of the cardiac cycle. All right, so how do we get our heart sounds? So S1 and S2 are what we think of. They're the lub dubs. There's also S3 and S4. These are soft sounds. So you hear lub, dub, right? So it's lub, dub, lub, dub. Okay. This is when we have the sound, is when you have blood flowing into the ventricles and during atrial contraction. Those loud sounds, the first one are produced by the AV valves, and the second loud sound is produced by the semilunar valves. So the dubs, semilunar, and the lub is AV valves. I've put some links down here so that you can hear what happens, or what a good heartbeat sounds like, or what happens when a person has a pericarditis, and when a person has mitral valve prolapse. When a person has mitral valve prolapse, it's like a whispering sound throughout. You never have the break between the lub and the dub. And when someone has acute pericarditis, you're going to hear scratching. Okay? I hope that you will listen to those.